When you look at a classic biology textbook, the illustrations always make the inside of a cell look so, I don't know, peaceful.
Yeah, very orderly, like a perfectly choreographed factory.
Exactly. A signal goes off, a protein floats down, lands exactly where it needs to be, does its highly specific job, and just leaves.
Right. It definitely implies this sense of absolute efficiency. Like, there's no wasted movement in biology.
But then, you encounter a paper like the one we are unpacking today, and suddenly that perfect textbook illustration goes completely out the window.
Oh, absolutely. It's a completely different picture.
Welcome to today's deep dive. Our mission today is to completely rewrite our understanding of how our mitochondria divide. We are looking at a groundbreaking 2026 study from Nature Cell Biology, and the cellular landscape it describes is, it's far more chaotic, dynamic, and honestly way more fascinating than those old diagrams.
It really is. And the stakes for understanding this division, which is a process known as mitochondrial fission, are incredibly high.
Right, because we always call mitochondria the power plants of the cell. But this isn't just basic cellular housekeeping, is it?
No, not at all. Fission is absolutely essential for distributing energy where it's needed, for quality control, and just overall cell survival. If this division process breaks down entirely, it leads to embryonic lethality.
Wow. So it just stops before life even starts.
Exactly. And if it's even just slightly dysregulated later on, it is heavily linked to tumorigenesis in various cancers, serious cardiomyopathies in the heart, and severe neurodegenerative diseases like Alzheimer's.
So getting the cut right is literally a matter of life and death.
It is.
But what actually, physically cuts a mitochondrion in half?
Yeah.
Because for years, the conventional wisdom said that the main division protein, which is called DRP1, just parachuted down from the cellular fluid.
Right. The idea was it landed exactly on a designated fission site and snipped the organelle in two.
Like a guided missile.
Like a guided missile, yeah. But this new research proves DRP1 doesn't do that at all. It actually acts as what they call a mito-scanner.
A mito-scanner.
Yeah, it roams. It browses the mitochondrial surface in these complex, continuous spiral paths to find the perfect spot to cut.
It's a total paradigm shift. Instead of a targeted missile strike, DRP1 is essentially a roaming surveillance system.
That's a great way to put it.
I was trying to picture this, and I keep coming back to a lumberjack looking to chop down a massive miles-long tree. You wouldn't just parachute a lumberjack into a random spot in the forest and hope they hit the right mark.
No, you'd miss the weak points entirely.
Right. You'd drop them on the trunk, and they would walk the length of the tree, inspecting the bark, actively looking for a weak point before they start swinging the axe.
And DRP1 seems to be our cellular lumberjack.
So today, for everyone listening, we're going to look at exactly how it walks, what it looks for, and who its molecular co-pilots are.
To understand how this cellular lumberjack operates, we first need to understand its tools. Like, what actually is DRP1?
Right.
At its core, it is a large GTPase.
Okay, let's unpack that term for a second.
Sure. So a GTPase is an enzyme that binds and hydrolyzes GTP. And GTP is a close cousin of ATP, which most people know as the cell's main energy currency.
The burning fuel.
Exactly. When DRP1 hydrolyzes or burns that GTP, it provides the energy for massive conformational changes. It basically forces the protein to violently change its physical shape.
So it's essentially got a built-in energy-burning motor.
That's a good way to look at it. But it's more of a multi-tool. The DRP1 protein actually has 4 main domains.
Okay.
There is that motor we just talked about. Then there is the central stalk, which acts like molecular Velcro, allowing multiple DRP1 proteins to stick together and self-assemble into large teams, or oligomers.
Oh, so they squat up?
They absolutely squat up. And connecting the motor and the Velcro is something called the bundle signaling element. Think of that like a transmission cable that passes the mechanical force from the motor down to the stalk.
Makes sense. Motor, transmission, Velcro, what's a fourth?
Finally, there's the variable domain, which acts as a sensor to feel the curvature of the membrane and help the whole complex attach to the mitochondria.
Okay, I can picture the multi-tool, but how did the researchers actually see this invisible lumberjack team moving inside a living cell? I mean, we aren't just looking at a static photograph here, right?
No, not at all. They had to use highly advanced super-resolution microscopy, specifically 2D and 3D structured elimination microscopy, or SIM.
Okay.
They took living cells and tagged the DRP1 proteins with a fluorescent green marker. And when they looked through the microscope, they didn't just see static dots. They saw these glowing green squads forming highly modal, spiral-like trajectories, actively moving along the length of the mitochondria.
But wait, how do you mathematically measure movement on a mitochondrion? I mean, they aren't stiff pipes. They are living, breathing, wiggly noodles floating inside the cell.
Yeah, they're incredibly dynamic.
So if the noodle is moving and the green dot is moving on the noodle, how do you track just the dot?
That was actually one of the biggest technical hurdles of the whole study, and their solution was brilliant. The researchers developed a mathematical tubular model.
A tubular model?
Yeah, they essentially wrote a computer algorithm to track the center of mass of the glowing DRP1 particles. Then they mapped the 3D position using cylindrical coordinates.
Okay, so wrapping around it.
Right. They plotted the length of the mitochondrial tube as the z-axis, measured the radius of the tube, and calculated the perpendicular angle wrapping around it. By doing this, the algorithm could mathematically subtract the wiggling of the organelle itself.
That is mind-blowing. They basically computationally unbent the noodle so they could isolate just the movement of the protein.
Precisely. It's a huge achievement. And once they had those clean, unbent coordinates, they calculated something called the mean square displacement over time.
Okay, what does that tell us?
For trajectories lasting longer than 15 seconds, they plotted how far, on average, the particle wandered from its starting point. And they found a strictly linear increase in displacement over time.
Hold on, I want to stop you right there. Because if the displacement increases linearly over time, that describes free diffusion.
That's correct.
But that completely ruins my lumberjack analogy.
Oh, I know.
A lumberjack walks down the tree with purpose, looking for a specific mark. If DRP1 moves via free diffusion, it doesn't have a tiny motor driving it down a track. It's not walking with purpose. You're telling me this critical, life-saving protein is just a blindfolded lumberjack wandering aimlessly in the dark.
It sounds completely counterintuitive, I agree. But yes, it relies on a purely stochastic or mathematically random search. There is no active transport pulling it along.
Wow.
It is driven purely by ambient thermal energy, randomly sliding and diffusing in a confined space along the 2D surface of the organelle. It has no idea where it's going, and that is exactly why it needs to move constantly. It covers ground through relentless random patrolling.
That is wild. Okay, so we have a blindfolded squad randomly sliding around. But does the squad at least stay together? Because I know some cellular proteins move by a process called treadmilling, where they drop pieces off the back and add new pieces to the front, kind of like a tank tread.
Right, they constantly recycle parts.
Yeah. So is our squad swapping out members as it wanders?
To test that, they performed a really elegant experiment using a photoconvertible tag called EOS. It's a protein tag that normally glows green, but if you blast it with a very specific UV laser...
Oh, I've seen this. It permanently shifts its structure and starts glowing red, right?
Exactly. So they hit the mitochondria with the UV laser, which turns all the DRP1 currently sitting on the surface red, but it leaves all the extra DRP1 floating in the cellular fluid green.
Got it. So you have a red squad on the surface surrounded by a sea of green floating nearby.
Right. They blasted it and then watched the red squads for over 200 seconds. Now, if DRP1 was treadmilling, picking up new green pieces from the fluid to move forward and dropping red pieces behind, you would see green fluorescence rapidly mixing into the moving red particles.
And let me guess, they didn't mix.
Not at all. The red particles stayed purely red. The ratio of red to green remained completely flat the entire time they tracked them.
Okay, so I want to do a quick recap here, just to make sure you listening have this whole picture straight in your head before we move on. First, mitochondrial division relies on DRP1, which we now know scans the surface instead of just landing on a target.
Right, that's point one.
Second, it moves via free diffusion, meaning it's a random but constant blind patrol. And third, these guys travel as stable teams, oligomers, without swapping out members.
Exactly.
Which brings up a massive question. Imagine you are patrolling a massive, miles-long area completely randomly with a blindfold on. What kind of physical cue would you possibly need to finally tell you to stop walking and swing the axe?
And that brings us perfectly to the actual geometry of the situation. We know the squad is stable, but to understand how they know when to stop, we need to know their physical shape.
Okay, what do they look like?
For this, the researchers had to use an even more powerful tool called STID microscopy.
Right, stimulated emission depletion.
Yes, which lets us see structures down to the nanometer scale. They found that these DRP1 squads mostly form two distinct shapes on the membrane: rigid lines and curved arcs.
I was looking at the measurements in the paper, and the exact dimensions of those shapes seem to be the key to the whole mystery. The STED data show that the lines are about 390 nanometers long, and the curved arcs are about 900 nanometers long.
Yes. And if we connect those measurements to the bigger picture of the cell, we run into a major physical problem.
Which is?
A normal, healthy, unconstricted mitochondrion has a perimeter, so the distance all the way around the outside of the tube, of about 1,500 nanometers.
Wait, if the arc is 900 nanometers long, but the tube it's sitting on is 1,500 nanometers around, the math doesn't work out. The arc can't possibly reach all the way around the tube.
Exactly.
A lumberjack's arms are literally too short to hug the tree. So how does it ever manage to wrap around and cut it?
It can't do it alone. It requires external help. You see, the mitochondria don't exist in a vacuum. They are surrounded by a dense jungle of other cellular structures. Most notably, a network called the endoplasmic reticulum, or ER, and cables made of actin filaments.
Okay, and what do?
These structures act like a tiny molecular lasso. Before DRP1 can ever do its job, the ER and actin physically squeeze the mitochondrion from the outside, pre-constricting it to create a narrow bottleneck. They squeeze it down to a diameter of about 180 nanometers.
Oh, I see. And 180 nanometer diameter means a drastically smaller perimeter. So suddenly that 900 nanometer DRP1 arc is more than long enough. It actually can reach all the way around.
You've got it. As the stable DRP1 arc diffuses randomly along the surface, blindly feeling its way, it eventually hits one of these narrow bottlenecks. And suddenly, the geometry matches perfectly.
Wow.
The arc can finally wrap around the tube, allowing the head of the protein to interact with its tail. And when the head meets the tail, it causes the entire particle to physically stall its diffusive motion.
So it stops walking.
Right. It condenses, it begins to rapidly rotate around the tight neck, it fires that energy-burning motor we talked about, and it initiates full fission, slicing the organelle in two.
The aftermath of that cut is one of the coolest details in the study, I think. After the snip happens, the DRP1 particle splits roughly equally in half.
Yeah.
The remaining pieces stay at the newly cut tips of the two new daughter mitochondria, and they just sit there, rotating continuously. I was trying to think of a way to describe this, and it feels exactly like dropping a rigid plastic bracelet down a tapered traffic cone.
That's a great visual.
Yeah, right. The bracelet slides freely down the top of the cone until the cone gets too wide. Or, in the case of our mitochondria, the protein slides freely until the organelle gets too narrow, and then it jams perfectly into place. It becomes what chemists call kinetically trapped by the sharp curve of the membrane.
That is a phenomenal analogy. The stall isn't triggered by some complex chemical signal telling it to stop. It is entirely dictated by the physical curvature of the membrane, finally matching the physical stiffness of the DRP1 polymer.
So to do our second recap here, synthesizing this geometry part.
Yeah.
1. Our blindly wandering squads exist as 900 nanometer arcs that are geometrically incapable of encircling a normal mitochondrial tube.
Correct.
2. They have to wait for the ER and actin to act like a lasso and pre-squeeze the tube down to a tiny bottleneck.
Yes.
And 3. As our squads randomly patrol, they bump into this tight spot, wrap around it, stall out like that bracelet jammed on a cone, and that physical jam triggers the division. But that leaves me with another question for you, the listener. If DRP1 is just a piece of plastic sliding down a cone, what pulls it onto the cone in the first place? And what dictates how fast it slides?
Well, to figure out what controls the sliding, the researchers turned to a digital sandbox. They built a highly sophisticated computer simulation called a coarse-grained molecular dynamics model.
CGMD.
Exactly. Essentially, they coded a virtual mitochondrion and a virtual DRP1 polymer to see if the laws of physics actually supported what they were seeing in the living cells. And they tweaked two specific variables in the simulation. First, the stiffness of the DRP1 polymer itself, and second, how strongly it binds to the surface.
And what they found for stiffness wasn't just a simple straight line where stiffer equals faster, right?
No, it was a U-shaped curve, a non-monotonic relationship. They found that if the DRP1 polymer was too flexible, it just bunched up into a compact ball. It diffused very rapidly, but it could never form that rigid ring needed to wrap the tube.
Like pushing a wet noodle.
Right. But on the other hand, if it was too stiff, it became a rigid rod that barely moved at all.
So it requires a Goldilocks zone. It has to be just stiff enough to hold an arc shape, but flexible enough to glide smoothly along the curved surface.
Exactly.
But what about the binding strength? Because DRP1 doesn't magically stick to the slippery lipid membrane of the mitochondrion all by itself, does it? It needs receptor proteins to act as adapters.
It does. It needs molecular accomplices. And the researchers meticulously tested the 4 known adapter proteins to see who was actually helping our lumberjack.
Who are the suspects?
Looking strictly at the data, they found each plays a very distinct role. The first one is called MFF. This protein is the initial recruiter. When researchers genetically removed MFF entirely, or tweaked DRP1 so it couldn't bind to MFF, DRP1 just floated uselessly in the cellular fluid.
Oh, wow.
Yeah, it never even landed on the mitochondria. And obviously, no landing means no scanning.
So MFF is basically the bouncer at the club. If you don't interact with MFF, you aren't getting on the dance floor.
Very much.
But what about the others?
Well, there's one called FIS1, but the data here was surprisingly clear. Knocking down FIS1 had almost zero effect on the scanning process. It seems to be more of a bystander in this specific motion.
Okay, so we can ignore FIS1 for the movement part.
Right, but then we have MID49 and MID51. These 2 are the essential co-pilots. Through some intense cross-correlation analysis, the researchers proved that DRP1 actually co-diffuses with them.
Co-diffuses.
Yeah, they literally form a complex, holding hands, and move together as a single unit across the membrane.
Okay, so MFF acts as the landing pad, but MID49 and MID51 actually get in the car and drive with the squad.
Exactly.
And the researchers proved exactly why this hand-holding matters by breaking the system, didn't they?
They did. They introduced specific genetic mutations into DRP1. So when they altered the protein so it struggled to hold hands with its MID49 and MID51 co-pilots, the scanning process ground to a halt.
It just stopped walking.
Yeah. And interestingly, they also tested a mutation that actually enhanced its binding to the MFF recruiter, essentially gluing it too tightly to the landing pad. In that case, too, the scanning speed severely slowed down.
It's all about balance, the Goldilocks zone again. But what I want to know is, what does this all actually mean for the cell? I mean, if the scanning slows down because the co-pilots are missing or the glue is too sticky, what is the consequence? Why is the walking speed so critical?
Because mitochondrial division is ultimately a numbers game. If the scanning slows down, DRP1 takes way too long to wander around and find those pre-constricted bottlenecks.
Right.
As a result, the number of successful fission events absolutely plummets. And when mitochondria aren't getting divided, they just keep fusing together. The result, which the researchers saw clearly under the microscope, is a massive elongation of the mitochondria.
They just turn into long spaghetti.
Exactly. You get this hyperfused, tangled network of giant mitochondria. A failure in motility, a slow walking speed, leads directly to a catastrophic failure in the organelle shape.
It's incredible. It proves that this random stochastic search is the actual rate-limiting step for the entire process. If the surveillance patrol is too slow, the whole factory breaks down.
Precisely.
So let's do our final recap to wrap our heads around this last piece of the puzzle. First, DRP1 requires a perfect Goldilocks balance of stiffness to scan efficiently.
Yes.
Second, MFF acts as the essential landing pad to pull it out of the cellular fluid. And third, MID49 and MID51 act as the co-pilots, locking in to travel the surface. If you break that bond, the scan stops, and the mitochondrial network gets tangled into spaghetti.
That covers it.
So think about that for a second if you're listening. Have you ever considered that the health of your entire body and your ability to stave off diseases like cancer or Alzheimer's might rely entirely on the literal walking speed of microscopic proteins?
It is a profound realization. If we synthesize the big picture from this paper, it fundamentally reframes how we view mitochondrial division.
Right, it's not just a chop.
Exactly. It isn't just an isolated chop that happens instantly on command. It is a highly regulated, 3-step sequential process. Step 1 is browsing that fast random scanning. Step 2 is sensing where the specific geometry of the DRP1 arc physically jams onto the pinch DR and actin bottlenecks. And step 3 is execution, the actual burning of energy to fuel the fission.
And this totally changes the game for how researchers have to look at metabolic diseases. If you're a scientist looking to cure cardiomyopathies or neurodegeneration, you can't just look at a cell and ask, is the DRP1 protein present? Or is the scissor broken?
No, you have to look for a broken scan. The speed of the search is the limiting factor for cellular health. What's most fascinating to me here is the broader implication of geometry acting as a fundamental biological regulator. We are so used to thinking of biology as a messy soup of chemical reactions, concentration gradients, and electrical signals.
But this paper suggests that the physical curves, The tight pinches and the literal shapes of a membrane act as a direct physical control mechanism. It's structural regulation.
It really is.
Which leaves us with a wild thought to mull over as we wrap up today's deep dive. We always visualize the cell as this liquid chemical factory. But what if your cells are actually shaped more like microscopic skate parks?
I love that.
What if the physical curves, ramps, and pinches dictate exactly where these molecular machines can travel, where they gain speed, and where they get trapped to do their jobs? What other critical life processes inside you are secretly just a matter of structural geometry?
It completely changes how you visualize life at the microscopic scale. It's a reminder to keep questioning our assumptions because clearly the textbooks are far from finished.
Remember that clean, precise textbook diagram we talked about at the beginning? We assumed everything just landed on its target like a guided strike. But the reality is so much murkier, much more chaotic, driven by random chance and geometry, and ultimately so much more beautifully engineered. Until next time on The Deep Dive, keep scanning.